Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course Developmental Biology. Today's lecture is Early Development in Frog. I am Dr. Priya Goyal, Assistant Professor, Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. This is the syllabus of Developmental Biology that we are following. We are currently doing Unit 2, Early Embryonic Development, in which we have already discussed gametogenesis, fertilization, planes and patterns of cleavage, various types of blastula, and fate maps in the previous lectures. So today we will do the early development of frog up to gastrulation. We will study gastrulation in general, different types of gastrulation movements, early development in xenopus, that is the African clawed frog up to gastrulation, fate map of frog, and we will also come across various terms like Gray Crescent, mid-blastula transition, portal cell, blastopore, cortical rotation, vegetal rotation, cauda mesodermal mantle, yolk plug, and few others. Let us begin with what is gastrulation in general. Louis Wolpert once said, "It is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life." In the previous lectures, we have discussed gametogenesis and then how male and female gametes fuse during fertilization to create a zygote. Then the zygote divides into a number of cells which first form a morula and then a blastula. Now, how a blastula is converted into a gastrula, in which there is formation of 3 germ layers, and how the cells of a blastula, that is, the blastomeres, arrange themselves with respect to each other. They change their shape, they change their size, they change their adhesion, they change their position with respect to each other. All these processes are involved in gastrulation. So, gastrulation is basically a set of coordinated cell movements, which involves changes in cell shape and cell adhesion. During gastrulation, blastomeres get rearranged to establish a multi-layered body plan. After gastrulation, the 3 germ layers, that is the outer ectoderm, the inner endoderm, and interstitial mesoderm, are laid down. Now, during gastrulation, the prospective endodermal and mesodermal cells are to be brought in the inside of the embryo, while the prospective ectodermal cells have to spread all over its outside surface. Gastrulation usually takes place by a combination of 2 or more type of formative and morphogenetic movements of the cells with respect to each other. And during gastrulation, cytoskeletal elements play an important role. They drive certain changes which help in cell shape change and cell adhesion changes. For example, microtubules generally elongate, which makes the cells columnar during gastrulation. Now, what are the various types of gastrulation movements that happen during gastrula formation? The first type is invagination. Invagination, as you see in the diagram, is a movement in which cells are invaginating or they are moving to the inside of the embryo from one side, as if somebody is poking a round tennis ball or a soft rubber ball from one end with its finger. Invagination is a movement which is seen during gastrulation in sea urchin endoderm. The second type of gastrulation movement is involution. As we see in the diagram, involution involves movement of an outer layer of cells onto its inner side. Now, the inner side of cells will form another layer which will keep on spreading on whatever cell layers are remaining on the outside. Involution is a movement which is seen during gastrulation in amphibian mesoderm. The third type of gastrulation movement is ingression. Ingression, as we see in the diagram, happens when the cells from an outer layer simply pull themselves off and they move to the interior of the gastrula, maybe in the blastocoele. Now, these ingressing cells generally form mesenchymal cells, that is, they remain free from each other. However, in certain other gastrula, they may come to the interior and they join some other layer of cell as a single layer. Ingression is seen during movement of sea urchin mesoderm and also in Drosophila neuroblasts. The fourth type of movement is delamination. Delamination happens when one layer of cell divides to form two layers. One layer will continue moving similar way while the other layer will move to the inside of the embryo and form another layer. Delamination happens during hypoblast formation in birds and mammals. The fifth type of gastrulation movement is epiboly. Epiboly predominantly occurs in the spreading of epithelial layers so as to form an outer covering all over the embryo. As we see in the diagram here, the cells present on the outside, which are predominantly the epithelial cells, they are spreading all over the inside by cell flattening, cell division, and also by intercalating several layers of cells into single layer. Epiboly occurs during ectoderm formation in sea urchins, tunicates, and amphibians. The sixth type of gastrulation movements are the intercalation movements, which involve wedging of cells between their neighboring cells. Intercalation can be a lateral one, which is also known as convergent extension, or a radial intercalation. The lateral intercalation involves lateral or the sideways movement of cells which happen within the same layer of cells. Lateral intercalation is an example in Xenopus where we see medio-lateral type of intercalation. Radial intercalation occurs perpendicular to the surface between adjacent layer of cells. It involves wedging of two different layers into one. This often leads to epiboly. During epiboly, as we discussed, the surface area of epithelium increases while its thickness will decrease, while it has to spread all over the outside of the embryo. Radial intercalation is seen in Xenopus marginal zone cells. Here we have an example to understand lateral intercalation on convergent extension. Here we have 5 cells in each row and 4 cells in each column. That is a total of 20 cells in which the upper 2 rows of cells are different and the lower 2 rows of cells are a different nature. Now, when lateral intercalation or convergent extension will happen, there will be 2 rows and 10 cells in each row will be present. Now, here we see that the cells have undergone intercalation within the same type of layers. That is, there is no intermixing between the layer of cells. So again, the total number of cells remains 20. However, the cells have undergone lateral increase in surface area, that is lateral intercalation or convergent extension between the same type of cells or the same type of layers. The second type of intercalation or the radial intercalation happens when the cells wedge between two different type of layers. For example, as we see here, in this diagram, there are 4 cells in each row and 2 cells in each column. When radial intercalation will happen between these two different type of cells, we will have a single row with 8 cells. Now we see that the cells have merged from 2 different layers to form a single layer. And now this single layer contains cells from both the different types here. During gastrulation, there are 3 crucial axes that lay the foundation of the body. The crucial axes, as shown here, are the anterior-posterior axis, which run from head to tail. The second is the dorsal-ventral axis, which run from the dorsal side, that is the back side of the body, to the ventral side or the side on towards the belly. The third is the right-left axis, which distinguishes between the right side of the body and the left side of the body. That is, it distinguishes between the two lateral halves of the body as we see here. Now let us begin with the early development in frog. Here we will take Xenopus, that is the African clawed frog, as a type study. Fertilization in amphibians is generally external. It happens in water after the male lays sperms over the eggs which have been laid by the female. The eggs may be laid in vicinity and then they approach each other, as we studied during the previous chapter of fertilization. The egg already has polarity when it is laid. It is meso-lacethal in nature. That is, it contains sufficient amount of yolk. Certain proteins and messenger RNAs are already localized in the unfertilized egg. Fertilization can occur anywhere in the animal hemisphere of the oocyte, while the point of sperm entry will determine the dorsal ventral polarity of the embryo. As we see here, this is an egg which has just been fertilized. The sperm has entered from this point. The sperm centriole has entered the oocyte or the ooplasm, and it has started formation of the ester rays and the microtubules so as to draw the female pronucleus towards itself. Now we see that the point of sperm entry will become the ventral side of the embryo, that is its belly side, while the side which is 180° opposite the point of sperm entry will become the dorsal side of the embryo. Now the axis which will be passing through this point of sperm entry to its directly opposite side will form the dorsal ventral axis of the embryo. Immediately after fertilization, there happens another phenomena which is known as cortical rotation in amphibians. Now students, an oocyte contains several pigment granules which are quite dark in color, which are located in the outer cortex of the cytoplasm in the upper half of the oocyte. They are not present in the lower half of the oocyte. Now this oocyte will look like a ball which is darker in the upper half and lighter in the lower half. Now immediately after fertilization, when the sperm will enter the oocyte, the upper cortex will undergo a 30° rotation. Now this 30° rotation with respect to the inner cytoplasm, that is, the inner cytoplasm will not rotate, just the outer cortex, this portion will rotate with respect to the inner cytoplasm. This rotation is known as cortical rotation. Now this cortical rotation might expose a region of light colored inner cytoplasm directly opposite the point of sperm entry in many amphibians. And this light colored area which has just been exposed is called the Gray Crescent. This Gray Crescent is the area or the region where gastrulation will actually begin. As we see what is the importance of formation of Gray Crescent or the cortical rotation here, we see that there are a number of transcription factors which were earlier present in the vegetal side of the oocyte. During cortical rotation, these transcription factors also shift towards the Gray Crescent. That is, they come to lie on the future dorsal side of the embryo where gastrulation has to begin. These transcription factors help in beginning of the gastrulation. There is a special role of microtubular arrays which have been organized by the sperm centriole in cortical rotation. Ultimately, we can say that cortical rotation determines the antero-posterior axis of the Xenopus embryo, since it places the transcription factor beta-catenin in the region opposite the sperm entry, that is the future dorsal region of the embryo, where it increases the expression of certain genes that initiate the involution of mesoderm, thereby establishing the antero-posterior axis of the embryo. Remember, the point of sperm entry determines the dorsoventral axis and the cortical rotation determines the antero-posterior axis of the Xenopus embryo. So after fertilization, a zygote is formed, and a zygote has to undergo the process of cleavage. The cleavage in amphibians is an unequal radial holoblastic type. As we know that the ovum in amphibians contains moderate amount of yolk, and most of the yolk is concentrated in the lower half or the vegetal half of the ovum. So this type of ovum is mesolacethal in nature. Yolk which is accumulated in the vegetal hemisphere will give a hindrance or impedance to cleavage. The first cleavage is meridional in nature, which occurs along the anteroposterior axis. And in species where Gray Crescent is present, it will bisect the Gray Crescent. The second cleavage is also meridional. It occurs at right angle to the first division. Now here one thing is important, that the first division starts to happen at the animal pole. It goes on a definite speed, but as it enters the vegetal half of the zygote, the speed of that cleavage furrow slows down because of the presence of large amount of yolk there. So before the first cleavage gets through the whole of the zygote, the second cleavage has already begun to happen. The third cleavage is latitudinal in nature. Latitudinal means slightly above equator. The same reason that yolk is present in the vegetal hemisphere, which impedes the formation of cleavage furrow through it, the third cleavage cannot be equatorial in nature but slightly above equator, latitudinal. This leads to the formation of 8 cells, in which the upper tier of cells are smaller in size, known as micromeres, while the lower tier of cells will be larger in size, known as macromeres. Now further cleavages will happen in both the micromeres as well as macromeres at the same rate until 12th cycle. Later, in the animal region, numerous small cells will be formed because the micromeres will divide at a little faster speed as compared to the macromeres, where more of yolk is present. Ultimately, a morula will be formed at 16 to 64 cell stage, and when it will develop a blastocele or a cavity, the stage is known as blastula, and blastula we say at 128 cell stage. Now blastula is the stage which has to enter gastrulation. This is a diagram which shows the fate map of amphibian. It also shows the cleavage of xenopus egg. Here also the first 3 cleavages are shown in the scanning electron micrograph. Now what are the functions of blastocele? Why is a blastocele formed in the blastula? So blastocele predominantly changes its shape during cell migration, and thus it allows the migration of cells during gastrulation. Also, it prevents the cells beneath it, that is from its floor, to interact prematurely with the cells lying at the roof of the blastocele. And if this premature interaction happens, it leads to some drastic changes in the gastrula. For example, if animal cap cells, which have to form ectoderm, come in contact with the vegetal region below the blastocele, they become mesoderm. What keeps the blastomeres together? It is the cadherins, predominantly the EP cadherin and numerous other cell adhesion molecules, which keep the cleaving blastomeres adhered together. The messenger RNA for this protein is supplied in the oocyte cytoplasm itself. This diagram shows the fate map of frog blastula. As we discussed during the lecture of fate map, all the blue colors indicate the prospective ectoderm, the red colors indicate the prospective mesoderm, while the yellow colors in the fate map indicate the endoderm. Now the exterior cells which are shown here always form the ectoderm or endoderm, whereas all the mesodermal cells are lying in the interior of the blastula. This is the blastopore, or the point at which the gastrulation will begin. The prospective ectoderm is the entire pigmented area which is present at the animal pole of the blastula. At the end of gastrulation, this ectoderm forms entire outer layer of the gastrula. Neural ectoderm on the future dorsal side of the embryo which develops into central nervous system as well as all the sense organs. The second type is the epidermal ectoderm, which is present on the anteroventral side. It forms the skin epidermis, nose, suckers, ear and ectoderm of mouth in the embryo. Prospective ectoderm is called the zone of expansion since it expands all over the outside of the embryo during gastrulation. Prospective mesoderm is formed as a crescentic pigmented zone or marginal zone present all along the inside of the endoderm near the equator of the blastula. Now this prospective endoderm consists of the notochordal part, which has to form the notochord and mesoderm of the frog embryo. It also consists of a prechordal plate, which is present in the form of a strip of cells beneath the notochord on the dorsal side. It forms the connective tissues of the embryo. Somatic mesoderm is present on the lateral sides. It helps in development of somites of the embryo. The lateral and ventral parts of the Gray Crescent develop into mesodermal lining of the body cavity, kidneys, heart, etc. The prospective mesoderm is called the zone of involution because the cells undergo involution during gastrulation. They involute to the inside of the embryo to form mesoderm. The prospective endoderm lies on the entire vegetal region of the blastula, which is the non-pigmented part of the blastula. Now, this prospective endoderm consists of an area just beneath the prechordal plate, which will form alimentary canal, that is the endodermal lining of the mouth, gill, and pharynx, while rest of the area develops into midgut, hindgut, liver, pancreas, urinary bladder, and endocrine glands. The prospective endoderm is called the zone of invagination because the endoderm undergoes invagination, gastrulation movements during the process of gastrulation. An important condition during gastrulation is the mid-blastula transition. During early cleavage, only a few genes appear to be transcribed until the 12-cell cycle. But now, when the embryo has to prepare for gastrulation, it needs to activate whole of its zygotic genome. That means the genes within each nucleus of the embryo have to become active. And it is this which is known as mid-blastula transition or the MBT. Now, during mid-blastula transition, different genes begin to be transcribed in different cells. The cell cycle acquires gap phases. Earlier, during cleavage, the gap phases were not present since the cells have to divide mitotically very quickly. The blastomeres acquire the capacity to become motile. That is, they have to move with respect to each other during gastrulation. Mid-blastula transition also involves some chromatin modification. Certain promoters are demethylated so as to allow transcription of the genes. For example, the vegetal cells under the direction of the veg-T protein, which is formed from the localized maternal messenger RNA, become the endoderm. And once the vegetal cells become the endoderm under the influence of veg-T protein, they start secreting factors that induce the cells lying above them, that is at the equatorial region, to become the mesoderm. Now, after mid-blastula transition, the gastrulation will begin. Where the gastrulation begins in xenopus? In the marginal zone or the equatorial region of the blastula. In the amphibian embryos, where Gray Crescent is also present, the region is precisely the Gray Crescent, where gastrulation will begin. This region is directly opposite the point of sperm entry. It marks the point where the animal and vegetal hemispheres meet because here the endodermal cells are not as large and yolky as they are in most of the vegetal area. The primary task of gastrulation being to bring the endodermal cells at the vegetal hemisphere towards the inside of the embryo. The ectodermal cells which were present in the animal hemisphere need to coat outside of the embryo during gastrulation, and the mesodermal cells have to be placed in between the ectoderm and the endoderm. They are predominantly the cells which will be found beneath the blastocele. This diagram shows the complete process of gastrulation in xenopus. We will discuss all these diagrams and stages one by one. So the first stage is invagination of bottle cells and the formation of blastopore. Gastrulation is first visible when a group of marginal endodermal cells on the future dorsal side of the embryo within Gray Crescent start invagination. As you see, this yellow zone shows the endodermal cells. Now, when a group of some marginal endodermal cells which are present on the future dorsal side of the embryo, they start invagination. We say that gastrulation has just begun. Now, these cells assume a characteristic bottle shape. That is, the neck of the cells is still attached to the outside of the embryo while their inner sides start to elongate. Thus, they have a long neck and a fat inner side, which is present on the Gray Crescent area of the embryo. And these cells with the characteristic bottle shape are then known as bottle cells. And these bottle cells start to form a slit-like blastopore, which is present here as shown here. These are the bottle cells. Here we see, and this is the formation of the blastopore or a crescent, or we can say an entrance of the cells toward the inside. This happens 19 hours post fertilization, and this shows the first stage in the formation of gastrula. This diagram shows the bottle cells here at the entrance of the blastopore, where an archenteron or the future gut has also started to begin. This diagram also shows the blastopore, which is a crescent-shaped structure present on the future dorsal side of the embryo. Now, before we begin further, we need to understand two important terms with respect to gastrulation of xenopus. The first is the non-involuting marginal zone or the NIMZ. The non-involuting marginal zone or the equatorial zone, as it says, is a part of the ectoderm. It is shown blue here. This zone does not undergo involution. It tends to stay on the outside of the embryo and it undergoes convergent extension and also epiboly. That is, it tends to form a surrounding area of the embryo during gastrulation. The second term is involuting marginal zone or the IMZ. Now this IMZ consists of two cells. The first set of cells are the red cells shown here. This cluster of Z cells is the deep involuting marginal zone or the deep IMZ cells. These deep IMZ cells undergo involution to form the inner lining or the endoderm of the archenteron. As we will see in the next diagrams, archenteron is the future gut of the embryo. The second type of involuting marginal zone cells are these yellow cells which are present as a single line here. These cells are known as the superficial involuting marginal zone cells. Now these superficial IMZ cells will undergo involution to form the internal lining of the archenteron roof. And both these, the red cells or the deep IMZ cells and the superficial IMZ cells together form the involuting marginal zone. So we see that the non-involuting marginal zone cells are the ones which are part of the ectoderm. They do not undergo involution. They stay on the outside of the embryo and they extend by convergent extension and epiboly. By the end of gastrulation, they spread all over the embryo. The involuting marginal zone cells include the deep IMZ cells as well as the superficial IMZ cells which form parts of the archenteron. The next step during gastrulation is the vegetal rotation and formation of archenteron. During the beginning of gastrulation, there is formation of the involuting marginal zone as we just discussed, which consists of the prospective head mesodermal cells shown pink here and the chorda mesodermal cells which are shown red here. Now there are large number of gastrulation movements in the presumptive endodermal cells. The pharyngeal endodermal cells which are shown orange here, they are placed first laterally to the moving blastocoele. And with more gastrular movements, these pharyngeal endodermal cells, they are displaced more toward the animal cap. And these pharyngeal endodermal cells are always placed immediately above these pink and red or the involuting mesodermal cells. As the bottle cells undergo more and more invagination, they lead to the formation of this archenteron, which is the primitive gut or gastrocele. It forms another cavity in the embryo. As archenteron expands on one side, the blastocele is gradually decreasing and is being displaced to the ventral side of the embryo. As it is being displaced, it gradually shortens in size. Finally, it is obliterated or lost from the gastrula. As archenteron deepens, its leading edge is formed of the bottle cells, which are now called the respread bottle cells. These bottle cells later on become the pharyngeal cells of the foregut, while the roof of the archenteron is formed by endoderm, which is derived from the superficial layer of the involuting marginal zone cells. Meanwhile, the lateral marginal cells involute over the lateral lip of the blastopore, and they become internal, that is, they come to lie on the inside of the embryo. In the vegetal region of the embryo, the large yolky cells also move inward, but they come to lie toward the posterior part of the archenteron floor. And remember, once the gastrulation is initiated, there is no more a need of bottle cells. There are certain experiments which have shown that if bottle cells are removed after the initiation of gastrulation, it does not affect any further gastrulation movements in the embryo. The third step during gastrulation is the involution of marginal zone cells or the mesodermal cells. As we know, the presumptive mesodermal cells are present in the deep marginal zone, while the presumptive endodermal cells lie in the superficial marginal zone of the embryo. Now, at the dorsal lip of the blastopore, the migrating marginal cells will always turn inward, and they travel along the inside of the animal hemisphere by involution movements. Now, the first cells to move through the blastopore are the pharyngeal endodermal cells of the foregut, as we see the orange cells shown here. The second cells to enter through the blastopore are the prechordal plate cells, which are the pink cells shown here. They are the precursors of head mesoderm, that is, they become part of the archenteron root. The third cells to involute are the chorda mesodermal cells, or the red cells, which are shown here. The red cells are the precursors of notochord. They form the mid-dorsal strip of the archenteron between the ectoderm and endoderm. And these chorda mesodermal cells are derived from the superficial layer of the involuting marginal zone. The somatic and ventrolateral mesoderm involute by rolling over the lateral and ventral lips of the blastopore. After mesodermal involution, that is, after all the mesodermal cells have undergone involution, they will move from posterior side of the embryo, that is, from the blastopore, towards the anterior end of the gastrula by forming a single layer of cells. And this single layer is known as chorda mesodermal mantle. This chorda mesodermal mantle is supposed to cover the entire space between the ectoderm and the endoderm, just leaving a small part at the anterior end of the gastrula, which will form the mouth later on. The fourth process during gastrulation in xenopus is the formation of yolk plug and its internalization. Actually, this is a process which is going on side by side during the previous three steps also. Now we know that the dorsal lip of blastopore is formed first of all. After the dorsal lip is formed, as is shown in the B diagram here, by the invagination of bottle cells, as we discussed in the first step of gastrulation, this dorsal lip starts to expand laterally and also ventrally. Now, as it develops these lateral lips and ventral lips, some additional mesodermal and endodermal precursor cells are able to pass through the blastopore to the interior of the developing gastrula. During this, the shape of the blastopore will change from crescent, that is its dorsal lip, to its horseshoe shape, that is when the dorsal lip also gets surrounded by the lateral lips, to the circular type, when all the dorsal lip, the lateral lips as well as the ventral lip of the blastopore have been formed. And this whole sequence takes about 7 hours. After this complete circular shape of the blastopore is formed, it develops a sort of ring all around the endodermal cells. As we know that endodermal cells are shown yellow here, and they are all moving to the inside of the embryo, while the blue colored ectodermal cells are covering a spread over the entire embryo. By this time, most of the endodermal cells have moved to the inside of the embryo. There is only a small patch of endodermal cells which are left on the outside, surrounded by blastoporal lips on all the sides. And this patch of endodermal cells which is visible through the blastopore lip is known as the yolk plug. And eventually, as gastrulation proceeds, the ectoderm will internalize whole of this endoderm also. The next phase during gastrulation involves the convergent extension of the dorsal mesoderm. Initially, the involuting marginal zone is several layers thick, but shortly before their involution through the blastopore lip, these several layers of the deep INZ cells which are present at the dorsal region of the embryo start undergoing radial intercalation. Ultimately, they form one thin broad layer. This intercalation further extends the involuting marginal zone towards the vegetal side of the embryo. The superficial INZ cells spread out by dividing and flattening, and along with the bottle cells, they are passively pulled toward the animal pole, where they form the endodermal roof of this archenteron. Meanwhile, the deep INZ cells reach the blastopore lip. They start undergoing involution into the embryo and initiate convergent extension along the medio-lateral axis, thereby integrating several mesodermal layers to form a long, narrow band which migrate toward the animal cap. Thus, the involuting marginal zone extends vegetally. As we see here, the NIMZ or the non-involuting marginal zone blue as well as the involuting marginal zone showing red here are spreading by convergent extension. The radial and mediolateral intercalations of the deep INZ cells actually are responsible for leading the continued movement of mesoderm to the inside of the embryo. So let us have a look at the factors which cause convergent extension. The first factor is the polarized cell cohesion, in which the involuted mesodermal cells will send out protrusions so as to contact one another. The second important factor is the differential cell cohesion, which requires an extracellular fibronectin matrix as well as adhesion molecules, for example, the paraxial and axial protocoadherins, which are begin to be transcribed in the paraxial and axial mesoderm respectively. Convergent extension also requires a calcium flux for contraction of the actin microfilaments that are responsible to cause these kind of intercalating movements. Along with this, the ectodermal cells undergo epiboly during gastrulation. Now, the cells which are present at the animal cap, which are the prospective ectodermal cells as well as the non-involuting marginal zone cells, they undergo expansion and spread by epiboly so as to surround the entire outside of the embryo, and thus they help form the surface ectoderm. This epiboly involves increase in cell number by cell divisions, cell flattening in the superficial layer of the cells, as well as intercalation of several deep layers into one by radial intercalation. It also requires an assembly of fibronectin into fibrils. Thus, the ectodermal cells are able to form a complete layer all around the deep embryo. Meanwhile, the blastopore is shifted to posterior surface of the embryo and it contracts to withdraw the yolk plug completely into its interior. The blastopore is reduced to a small slit which is called proctodium. This proctodium joins the endodermal outgrowth to form the cloacal aperture of the embryo, and this marks the termination of gastrulation. So, what is the net outcome of gastrulation? After gastrulation, the endoderm comes to lie on the interior of the gastrula. Here, as we see, all of the yellow cells have come to the interior of this gastrula. The endoderm is derived from the superficial INZ cells or the involuting marginal zone cells, which forms the roof of the archenteron, and the yolky blastomeres, which are present on the vegetal half of the blastula, which form the floor of the archenteron. The ectoderm develops all the layer on the outside of the embryo, that is on its outside surface, while the mesoderm comes to lie in between the ectoderm and the endoderm. So let us discuss the lecture in a summary. What is gastrulation? It is a series of coordinated cell movements which involve changes in cell shape and cell adhesion to form a three-layered embryo. The point of sperm entry determines the dorsoventral axis. That is, the point at which sperm enters becomes the ventral side of the developing embryo, while the side 180-degree opposite to it forms the dorsal side. And another phenomena, cortical rotation, determines the antero-posterior axis of the xenopus rotation, which also shows the Gray Crescent area being developed. Though the Gray Crescent is not formed in xenopus, it is formed in many other amphibian embryos. Cleavage in xenopus is radially symmetrical, unequal, and holoblastic. Where gastrulation begins, it begins in the marginal zone in the region of the Gray Crescent if it is developed. The blastopore appears as the first sign of gastrulation, at which a large number of cells form a specific shape of bottle, and hence they are known as bottle cells. They start to invaginate, and this marks the future dorsal side of the embryo. Various cell adhesion molecules like E-peak adherins keep the cleaving blastomeres together. Ectoderm forms the precursor of the epidermis, brain, and nerves, is found on the outside of the embryo after gastrulation. Endoderm, which is the precursor of the lining of the gut and respiratory systems, comes to lie deep inside the embryo. Mesoderm, which is the precursor of the connective tissue, muscle, blood, heart, skeleton, gonads, and kidneys, or we say most of the organs of the body, comes to lie in between the ectoderm and the endoderm. So after a thorough discussion of the process of early development up to gastrulation in frog embryo, let us think over certain questions. Highlight the main features of amphibian gastrulation, in which you can discuss all the main highlights that have occurred during the process of gastrulation in amphibian. What is the role of bottle cells here? You have to justify a statement. The role of bottle cells is restricted to the initiation of gastrulation process, but not to its continuation. Can you comment on the role of cytoplasmic factors in frog gastrulation? We have discussed a number of cytoplasmic factors in different slides, so you have to compile all of them if you are asked such kind of question. Then can you justify the statement, the cells constituting the lip of the blastopore are constantly changing? As we were discussing various steps of the gastrulation, we kept on thinking of the various type of cells, the pink cells, the red cells, or the orange cells, which were moving through the blastopore and moving inside of the embryo. So you have to list out all those cells which are moving through the lip of the blastopore one by one in this justification of this statement. What are the main forces which drive the elongation of the archenteron, whether they are the bottle cells, whether they are some kind of gastrulation movements, So you have to write down the name of the gastrulation movement here and the cells which drive the elongation of the archenteron or the future gut. Then is how are the changes in cell shape and motility during gastrulation dependent on the changes in cell surface properties such as adhesiveness to the substrate or intercellular interactions. So here you have to think of the changes in cell shape and motility that are occurring during gastrulation vis-a-vis the changes in the adhesiveness, whether some adherents were being produced at some time or some other intercellular interactions were taking place. So you have to derive an answer to this question on those lines. These are the books which have been referred for the preparation of this lecture. I hope you had a good time listening to me. Thank you. Happy learning.